We are now finally able to understand how tumors are able to trick our immune system to feed them with inflammation. When we're born and grow up, we have immune cells that attack everything but us, because the ones that recognized us were eliminated. Cancer cells may be mutated, but they're still our cells, so how do they get the immune system's attention? Presumably any B cell that would recognize us was killed off before our birth. That's where xenoautoantibodies come in. Xeno means foreign, as in xenophobia. Auto comes from the Greek autos, meaning self, as in automobile, a self-moving vehicle, no horse required. But how can something be both xeno and auto, foreign and self? Though many tumors, like breast cancer, thrive on inflammation, thrive on this low-grade immune response, the problem for the tumor is that since it originated from normal breast tissue, it doesn't look foreign to the immune system, so the tumor isn't able to evoke the response it needs and wants. That's where NuGC comes in. Evidence for a human-specific mechanism for diet and antibody-mediated inflammation in carcinoma, cancer progression. See, Neu5Gc incorporation by the cancer confers a selective advantage to tumor cells that are best at accumulating Neu5Gc. By actively accumulating this foreign meat and dairy molecule coming from our diet into the tumor, the cancer may use it to trick our immune system into producing autoimmune antibodies, anti-Neu5Gc autoantibodies, which leads to the release of inflammatory compounds that enable the cancer to grow faster. The combination of tumor-associated Neu5Gc and our circulating anti-Neu5Gc autoantibodies promotes tumor growth by inducing weak inflammation, causing infiltration of inflammatory cells, and enhancing angiogenesis, enhanced blood flow to the tumor. Our own tumors use what we feed on to get what they feed on. So, as I talked about before, since cancer, in a sense, may be now considered an autoimmune disease, what are the dietary implications, given the fact that most animals produce this tumor promoter? Well, we don't make Neu5Gc, at least we haven't for a few million years, and neither do plants. So to undermine our tumor's sneaky strategy, I guess it comes down to either cannibalism or a plant-based diet, or both, I guess. Importantly, these data are congruent with the decreased cancer risk associated with veganism. Probably about a third of common cancers can be prevented by eating a healthy plant-based diet, being physically active, and maintaining a healthy weight. One of the ways plants may help is by cutting off the supply lines to cancerous tumors. A tumor cannot grow without a blood supply. Currently, it's believed that a tumor mass cannot exist in a volume greater than about the size of the ball at the tip of a ballpoint pen without a proper blood supply, which indicates that angiogenesis, angio means vessel, so the genesis, the creation of new blood vessels, is critical to tumor growth. Each one of us has cancer cells in us right now. By age 70, microscopic cancers are detected in the thyroid glands of virtually everyone, for example. Most of these tumors never cause problems, never become clinically significant, leading to the concept of cancer without disease as a normal state during aging. Cancer cells are commonly present in the body, but they can't grow into tumors any bigger than that tiny dot size, no more than 10 million cancer cells, before needing to get hooked up to a blood supply. So, tumors diabolically release angiogenic factors, chemicals, that cause new blood vessels to sprout into the tumor. The most important one is called VEGF, vascular endothelial growth factor. But we can suppress VEGF with veggies. Many of the phytonutrients we know and love in tea and spices and fruit and berries and broccoli and beans can block cancer's stimulation of new blood vessels. They're ideal for prophylactic long-term use against breast cancer because of their reliability, availability, safety, and affordable price. Dietary agents used to suppress angiogenesis may be an important step in the prevention and treatment of breast cancer, and in fact, all types of tumors. The review concludes that we now have convincing evidence that dietary plant constituents possess the unique ability to affect tumor angiogenesis, which may be deemed advantageous in the prevention and treatment of human breast cancer. Most of these studies have only been done in a petri dish, though. You stimulate human blood vessel cells, and they start forming these tubular structures, trying to make new capillaries to feed the tumor. But if you add plant flavones like apigenin or luteolin found throughout the plant kingdom, like in citrus, celery, and peppers, you can see they help block the tube formation. Here's the effect of fisetin, a phytonutrient found in strawberries and other fruits and veggies. It just shrinks the beginnings of new blood vessel formation right on down. Where do researchers get their hands on human blood vessels? From human umbilical vein endothelial cells. They get them from discarded umbilical cords, or more controversially, from the eyes of aborted fetuses. But either way, you can stimulate blood vessel formation with the tumor compound VEGF, and then abolish that effect with plant compounds, in this case from purple rice. Therefore, the daily consumption of natural foods containing adequate flavonoids could be beneficial for the prevention of cancer metastasis, or could improve cancer prognosis. Given the power of plants, one might speculate that the foundation of an anti-angiogenic approach to cancer might be a whole food, vegan diet. Our story begins on a Norwegian fur farm in 1957. Mink were dropping dead left and right from a malignant new liver disease. The clue came when livestock started dying from liver cancer as well. What tied all the cases together was the use of fish meal in their diets, fish meal that the country had just started preserving with sodium nitrite. Subsequent research discovered nitrite, under certain circumstances, can form nitrosamines, which directly attack DNA, or universally condemned as one of the key carcinogens in cigarette smoke. The occurrence in food was raised as a matter of gravest concern nearly a half-century ago. Now we know the nitrites added to processed meats can form these carcinogenic nitrosamines, now recognized as among the most potent chemical carcinogens. For example, pregnant women who eat hot dogs risk having children with brain tumors, the number 2 pediatric cancer. Then children who eat lots of hot dogs have nearly 10 times the odds of developing childhood leukemia, the number 1 pediatric cancer. Last year in Meat Science, an article about the role of ham in a healthy diet breathed a sigh of relief. Aspects related to health and well-being are increasingly important factors in consumer decisions, although the great palatability of ham largely outweighs such considerations. The results of the recent aspirin meta-analysis suggesting a reduction of cancer mortality by about a third in subjects taking daily low-dose aspirin can justly be called astounding. Yet the protection from Western cancers enjoyed by those eating more traditional plant-centered diets is even more dramatic. Before the Westernization of their diets, animal products made up only about 5% or less of the Japanese diet. And here's their cancer rates compared to the United States at the time. Note that age-adjusted death rates from cancers of the colon, prostate, breast, and ovary were on the order of 5 to 10 times lower in Japan than in the US, with the mortality from pancreatic cancer, leukemias, and lymphomas 3 to 4-fold lower. But this phenomenon was by no means isolated to Japan. Western cancers were likewise comparatively rare in other societies where people ate plant-based diets. The cancer protection afforded by lifelong consumption of a plant-based diet, in conjunction with the leanness and insulin sensitivity that tends to come along with it, may be very substantial indeed. Therefore, a lifestyle protocol for minimizing cancer risk may include a whole food, plant-based diet. Now, if part of this cancer protection arises out of the aspirin phytonutrients in plants, are there any plants in particular that are packed with salicylates? Though salicylic acid, the main active ingredient in aspirin, is ubiquitously present in fruits and vegetables, herbs and spices contain the highest concentrations. Chili powder, paprika, turmeric have a lot, but cumin is like 1% aspirin by weight. Eating a teaspoon of cumin is like taking a baby aspirin. Consequently, populations that incorporate substantial amounts of spices in foods may have markedly higher daily intakes of salicylates. Indeed, it's been suggested that the low incidence of colorectal cancer among Indian populations may be ascribed in part to high exposure to dietary salicylates throughout life from spice consumption. The population in rural India has one of the lowest rates of colorectal cancer in the world. They have a diet that could be extremely rich in salicylic acid, given the substantial amounts of plant foods flavored with large quantities of herbs and spices. Some have proposed it's the curcumin in the spice turmeric, but maybe it's the salicylic acid in cumin, and the spicier the better. A spicy veggie vindaloo may have 4 times the salicylates of a mild Madras-style veggie dish. One meal, and you get a spike in your bloodstream like you just took an aspirin. So eating flavor-filled vegetarian meals with herbs and spices may be more chemoprotective, meaning more protective against cancer than just regular, more bland vegetarian meals. We may also want to eat organic. Because salicylic acid is a defense hormone of plants, the concentration is increased when plants become stressed, like when plants are bitten by bugs, unlike pesticide-laden plants. And indeed, soups made from organic vegetables were found to have nearly 6 times more salicylic acid than soups prepared from conventionally grown ingredients. We should also choose whole foods. Whole grains breads, which are high in salicylic acid, contain about 100 times more phytochemicals than white bread, 800 perhaps compared to 8. That does raise the question, though, what about the other 799? Interest in the potential beneficial effects of dietary salicylates has risen in part because of the extensive literature on the disease-preventative effects of aspirin. However, it should not be forgotten that plant products found to contain salicylic acid are generally rich sources of a whole long list of other phytonutrients, many of which have marked anti-inflammatory and antioxidant activity as well. Their potential protective effects should therefore not be overlooked. In this context, the importance of dietary salicylic acid should not perhaps be overemphasized. Indeed, some believe that salicylic acid deficiency has such important public health implications that it should be classed as an essential vitamin, namely vitamin S. But what they're really saying is that we should all just have to eat a lot of plants. According to a recent review, the most notable development in the epidemic of prostate enlargement and lower urinary tract symptoms is the recognition that modifiable lifestyle factors substantially influence the natural history of these conditions. These are the factors associated with increased risk: obesity, diabetes, meat, and fat. And some associated with decreased risk: exercise, moderate alcohol consumption, and vegetables. Which vegetables? Garlic and onions appear to help. I like how they call them onion users. Cooked vegetables appear to work better than raw, so maybe it's the carotenoids. And legumes were also found protective: peas, beans, and lentils. And flax seeds appear so powerful that they may be used to both prevent and treat the condition. A randomized, double-blind, placebo-controlled clinical trial that found that flax phytonutrients not only alleviate BPH symptoms, but their efficacy appeared comparable to the drugs we spend $1 billion on, without the side effects. Flax seeds also work against prostate cancer. These researchers had men who were about to get their prostates removed eat 3 tablespoons of flax seeds a day for the few weeks before surgery. They were skeptical that they would observe any differences in tumor biology between the flaxseed fortified diet-treated groups, patients, and the controls with such a short-term dietary intervention, but they found significantly lower cancer proliferation rates and significantly higher rates of apoptotic cancer cell death, cancer cell suicide I've talked about. Thus, these findings suggest that a flaxseed-supplemented low-fat diet may have an effect on prostate cancer biology. Just a few things have been found associated with a significantly increased risk for the disease: refined grains like white bread, also eggs, and poultry, which appeared even worse than red meat or desserts. Although these data are observational, more research is necessary. There's little, if any, downside to promotion of healthy lifestyle interventions: weight loss, exercise, decreasing meat and fat intakes, and increasing vegetable intake among those with prostate problems, particularly since these interventions possess proven benefits to overall and cardiovascular health. And how about before you're a prostate patient? Lifestyle habits associated with a decreased risk of developing clinical BPH are also highly likely to be beneficial in reducing the risk of erectile dysfunction, cardiovascular disease, diabetes. For this reason, urologists should encourage all men to undertake a prostate-healthy lifestyle. In addition to suppressing liver cancer growth in vitro, cranberries have often been found to have similar effects against human breast, colon, brain tumor, oral, and ovarian cancer cells. Here's the latest looking at prostate cancer cell growth. The United States has the highest rate of prostate cancer in the world, so let's try a Native American fruit. Researchers started out with about 50,000 human prostate cancer cells in a Petri dish, and if you do nothing, within a day you're closer to 100,000, then 200,000, then nearly 400,000 within 72 hours. But by adding just a smidgen of cranberries, or 2 smidgens, you can see they block that exponential cancer growth. The reason they tested such tiny concentrations is that we only absorb a small fraction of the cranberry phytonutrients we eat into our bloodstream. Still, cranberries are cheap. If drug companies and supplement manufacturers are going to capitalize on this, they need to define cranberries' active ingredient. Here's some of the various phytonutrients in cranberries, so different fractions were tested against various types of cancer to find the magic bullet. Yes, the anthocyanin phytonutrients inhibit colon cancer cell proliferation, about 15%, for example, about the same with the proanthocyanidin fraction, but nothing compared to the total cranberry extract of the whole fruit. There seemed to be an additive or synergistic anti-proliferative effects resulting from a combination of the various components compared to individual purified phytochemicals. So it's always better to eat the whole fruit. How do you do that with cranberries, though? Although 5% of cranberries are sold fresh, the vast majority are consumed as processed products. To get the same amount of anthocyanin phytonutrients in a cup of fresh or frozen cranberries, you'd have to drink 16 cups of cranberry juice cocktail, eat 7 cups of dried cranberries, or 26 cans of cranberry sauce. The problem is that raw cranberries are so tart that folks may opt for the dried. In a taste test survey, consumers said they wouldn't mind eating sweetened cranberries every day, whereas raw cranberries slope down towards maybe once a year. The problem is dried cranberries tend to come sweetened. Raw cranberries don't affect your blood sugar, but sweetened dried cranberries do, even the low-sugar varieties. What about cranberry juice, or shall I say, quote-unquote juice? Cranberry cocktail is only about a quarter cranberry juice. The ruby red phytonutrients in cranberries and pure cranberry juice are powerful antioxidants, increasing the antioxidant capacity of our bloodstream within hours of consumption. But the high fructose corn syrup acts as a pro-oxidant, even if you add vitamin C to it, as they did here, canceling out some of the cranberry benefit. So how do you get the upsides without the down? Check out my pink juice video, where I offer a recipe for making no-sugar-added whole fruit cranberry cocktail. Researchers showed that those at high risk for colon cancer could reverse the progression of their disease by taking curcumin, the yellow pigment in the spice turmeric, and curry powder, cutting down on precancerous lesions, and even precancerous lesions. Are there other high-risk lesions we can try spicing up? How about giving turmeric extracts to people who just had bladder cancer taken out, or have had early-stage squamous cell carcinoma skin cancer caused by arsenic exposure, or early-stage cervical cancer, or precancerous lesions in the mouth or the stomach? In about a quarter of the patients, the lesions started to get better. 1 out of the 2 bladder cancer survivors, 2 out of 7 precancerous mouth lesions, 1 out of 6 precancerous stomach lesions, 1 out of the 4 early-stage cervical cancer cases, and 2 out of 6 early-stage skin cancer, all without noticeable side effects. One of the reasons it may work better in some cancers than others, or some people better than others, is differences in bioavailability. Megadoses were given, and just a tiny amount ended up in their bloodstream. If you're treating skin cancer, though, why not just put the curcumin directly on the skin? I've talked about what turmeric compounds can do to cancer cells in a petri dish. Here's some before and after pics. These are cervical cancer cells in a petri dish with more and more curcumin. Normal cells are unharmed, but cancer cells are laid to waste. Yeah, but to make it to the cervix, curcumin must be absorbed unless a vaginal cream was invented. See, a variety of delivery methods have now been devised. Besides intravaginal, there's also oral, intra-abdominal, intramuscular, under-the-skin injection, straight into the veins or the arteries, on the skin, up the bladder and the nose, breathed in like an inhaler, up where the sun don't shine, or straight into the spinal column, bone marrow, the tumor itself, or implanted somehow. Taken orally, some of it does actually get into the tissue. You can measure the amount of curcumin absorbed into the wall of the intestine by examining biopsies and surgical specimens taken after a curcumin regimen. It makes sense to take turmeric orally to treat colon cancer, but if we have cancer erupting on our skin, why not just rub it on directly? And that's what these researchers did. Turmeric and curcumin as topical agents in cancer therapy. They just took some turmeric from the store, made a tincture out of it, dried it, put it in Vaseline, and then had cancer patients rub it on their cancer 3 times a day. What kind of cancer can you get at with a finger? These were folks with cancers of the mouth, breast, skin, vulva, and elsewhere. Isn't breast cancer under the skin? Not always. Advanced breast cancer can ulcerate right through the skin. And these were all people with recurrent ulcerating tumors that had failed to respond to surgery, radiation, and chemo. And these open cancers can stink and itch and ooze. There was nothing else modern medicine had to offer. So let's rub on some turmeric ointment and see what happens. It produced remarkable relief. A reduction in smell was noted in 90% of the cases, even in extensively ulcerated cases of breast cancer, and a reduction in itching in almost all cases as well, for example, relieving the severe itching in 2 of the vulva cancer patients. Most of the lesions dried up, and in many cases this relief lasted for months, all from just rubbing on some harmless spice turmeric, which they describe as an indigenous drug, highly effective in reducing smell, itching, and exudate. The effect of the so-called drug is remarkable, and that drug is just some edible spice used in curries for centuries. The pomegranate has been revered through the ages for its medicinal properties. So much so it's been used as a symbol for some medical organizations. A fruit seems to me a better representation of health than a snake on a stick. Supposedly beneficial for a wide range of diseases. Even the cannibals love it, improving the color of kid meat. No, they're talking about baby goats. It just gave me a bit of a double take when I saw this study. Most of the attention over the last decade has focused on pomegranates and prostate cancer, starting with in vitro studies, showing more and more pomegranate extract can suppress the growth of prostate cancer cells in a petri dish by up to 95%. Well, what about normal prostate cells? This is what normal prostate cells look like under a microscope with a little or a lot of pomegranate extract. No real difference. Doesn't seem to do much to healthy cells, but here's what prostate cancer looks like, just decimated by the pomegranate. But this was in a petri dish, not a person. Yes, if these results translated to the clinic, it could be dramatic, but first we have to try it out in people. Primary management of prostate cancer consists of either radical surgery or radiation. Now, despite this, a significant number of patients relapse and ultimately develop metastatic disease. Even after a radical prostatectomy, in about a third of the patients, the cancer comes back, as evidenced by rising PSA levels. At that point, the treatment options are limited. You already took out the prostate. The next step is essentially chemical castration, hormonal ablation. Just like breast cancer can thrive on estrogen, prostate cancer can thrive on testosterone. So you can try to wipe out testosterone, but that can have such negative side effects. Anything we can do to delay that would be good. So what about plants? They note that men in Asia have the lowest prostate cancer rates in the world, up to 10 times less than North America. Now, is this just a genetic thing? No. When Japanese individuals move to the United States and start eating and living like us, their breast and prostate cancer rates shoot up right towards ours. This could be because of what they're eating more of here. Animal products is the strongest risk factor for prostate cancer worldwide on kind of a country-by-country basis, or because of what they're eating less of here, their traditional low-fat, high-fiber diet, with soy products and green tea, and plant-rich in general. So, did these researchers try sticking these cancer patients on a plant-based diet? No, they just had them drink a cup of pomegranate juice every day. Why? Because the study was funded by the pomegranate juice folks. At least they were allowed to take it by mouth. So what happened? Well, in the 3 years leading up to the study, their cancer was steadily growing, as measured by their average PSA levels going up. Then once they started the juice, their tumors continued to grow, but it looks like they were growing slower. Now, in contrast, Dr. Dean Ornish and colleagues got an apparent reversal in early prostate cancer growth with a plant-based diet and other healthy lifestyle changes. PSA didn't just go up slower, it trended down. And dripping the blood of men on prostate cancer growing in a lab, the blood serum of those eating healthy suppressed cancer growth nearly 8 times better, whereas the blood of the men on the juice just suppressed cancer growth by about 12%. But still, to see anything just drinking a cup of juice every day is pretty impressive. The problem is there was no control group. Now, you could say they kind of acted as their own controls. This is them before and after. It's probably not just a coincidence that their tumors started growing slower right when they started the juice. But check this out. This is a drug trial trying to do the same thing. Treat men with recurring prostate cancer after surgery or radiation. In the drug group, tumor growth slowed in 55% of the men. Pretty effective drug, right? The sugar pill worked 73% of the time. The placebo effect can be so powerful that it may slow cancer growth. This is why we need placebo-controlled trials. Maybe tricking people into drinking pomegranate-flavored Kool-Aid would have the same effect, right? You think you're doing something for yourself, and so the cancer might slow. You don't know until you put it to the test. Finally, a randomized controlled trial of pomegranate juice for prostate cancer, and as you can read in the title, it had no impact. What do they mean, no impact? 25% of the cancer patients appeared to shrink their tumors as soon as they started drinking the pomegranate juice. Yeah, but 35% shrunk their tumors not drinking pomegranate juice. So any effect appears to just be a placebo. It's the same story with pomegranate extract pills. They seemed to work until they went head-to-head with sugar pills and fell flat on their face. Previously, I've explored the beneficial effects of flax seeds on prostate cancer, as well as breast cancer prevention and survival, thought to be because of the lignans in flax seeds, which are anti-cancer plant compounds found in red wine, whole grains, greens, cruciferous vegetables, but especially sesame seeds and flax seeds, the most concentrated source on Earth. But this is per unit weight. People eat a lot more grains than seeds. And of the grains people eat, the highest concentration of lignans are found in rye. So can rye intake decrease the risk of breast cancer? Theoretically, yes, but unlike flax seeds, it's never been put to the test. If you measure the level of lignans in the bloodstream of women living in a region where they eat lots of rye, the odds of breast cancer seem to be cut in half for women with the highest levels, but lignans are also found in tea and berries, and so they couldn't be sure where the protection was coming from. So researchers decided to measure alkyl resorcinol metabolites, a class of phytonutrients relatively unique to whole grains. They collected urine from women with breast cancer and women without, and those with breast cancer had significantly lower levels compared to women without breast cancer, be they omnivores or vegetarians. This suggests that women at risk for breast cancer consume significantly lower amounts of whole grains like rye. But if you follow older women in their 50s through 60s, the intake of whole grain products was not associated with risk of breast cancer. A similar result was found for older men. Whole grain consumption in one's 50s and 60s did not seem to be linked to prostate cancer risk. Is it just too late at that point? Well, we know from data on dairy that diet in our early life may be important in the development of prostate cancer, particularly around puberty when the prostate grows and matures. And so if you go back and look what men were drinking in their adolescence, daily milk consumption appeared to triple their risk of advanced prostate cancer later in life. And so researchers looked at daily rye bread consumption during adolescence, and indeed, those who consumed rye bread daily as kids may have cut their odds of advanced prostate cancer in half. This is consistent with immigrant studies suggesting that the first 2 decades of life may be most important for setting the pattern for cancer development in later life. Certainly important for how we should feed our kids, but if we're already middle-aged, is it too late to change course? To answer that question, researchers in Sweden put it to the test. How about we take men who already have prostate cancer, split them up into 2 groups, half get lots of rye bread, the other half get lots of high-fiber but low-lignan wheat bread, and see what happens. There's been some indirect evidence that rye may be active against prostate cancer, like lower cancer rates in regions with high rye consumption, but it has never been directly investigated until this study. Biopsies were taken from their tumors before and after 3 weeks of bread eating, and the number of cancer cells that were dying off were counted. There was no change in cancer cell clearance in the control bread group, but a 180% increase in the number of cancer cells being killed off in the rye group. A follow-up study lasting 6 weeks found a 14% decrease in PSA levels, a cancer marker suggesting a shrinkage of the tumor. Now, they note since they used very high rye bread intakes, it remains to be tested if more normal intake levels would have effects that are of clinical importance. As any good red-blooded American, my ignorance of the metric system did not flag 480 grams of rye bread a day as out of the ordinary, but that translates to 15 slices a day. Rather than eating a loaf of bread a day, the same amount of lignans can be found in a single teaspoon of ground flax seeds. We've covered how to prevent the first step of atherosclerosis, decrease the level of bad cholesterol in our blood. What about blocking some of these other steps downstream? Both common and specialty mushrooms inhibit adhesion molecule expression and in vitro binding of monocytes to human aortic endothelial cells in a pro-inflammatory environment. So that means mushrooms may help block both this step and this step. Basically, what these researchers at Arizona State did was take the lining of a human artery, soak it overnight with either nothing, the control group, or shiitake mushrooms, crimini mushrooms, oyster mushrooms, maitake mushrooms, or plain white button mushrooms. Then they took away the mushrooms, washed the artery off, and then added some monocytes before and after inflammation. So what we'd like to see is these bars come down, less monocyte adhesion. So instead of being sucked into the walls of our arteries, they can go off and do their business elsewhere. Which mushroom do you think worked the best? They all worked, but in another victory for this little fungi, plain old cheap white button mushrooms worked the best. And under inflammatory conditions, they found the same thing, but shiitake didn't even seem to work much at all. The health implications are that diverse mushrooms, including common and specialty mushrooms, can protect against cardiovascular disease by interfering with events that contribute to atherogenesis.
Thank you very much. So actually, I'm a medical physicist working at the University Hospital of Würzburg in Germany, and doing some research on ketogenic diet, together with my colleague Ulrike Kämmerer, who is advising patients on ketogenic diets for many years now. And together we founded the German Society for Paleonutrition recently, to provide a broader basis for all the people interested in the paleo lifestyle in Germany. Now, originally, I was supposed to give this talk together with my colleague Dirk Lemke. You can see him up here. And he should have spoken about the roles of exercise and vitamin D in cancer prevention and treatment, but unfortunately he was not able to come to this symposium, so I changed the title a little bit and will concentrate on the diet part today. So what I'm going to do is to relate certain aspects of a Paleolithic diet with biological mechanisms that play a role in cancer initiation and progression. I think most of you agree with me when I say that cancer can be considered a disease of civilization. And this notion is supported by reports from the beginning of last century, which stated that there was almost no cancer prevalence among primitive hunter-gatherer societies, including Eskimos, African tribes, American Indians, or tropical islanders. So this immediately leads us to ask, what has changed in our lifestyles that gave rise to the still increasing cancer rates? Most of you noticed already, and it was recently pointed out by Carrera Bastos et al. In a nice paper, that the main lifestyle factors in which our modern lifestyle differs from a more traditional Paleolithic lifestyle include exercise, sun exposure with the accompanying vitamin D status, chronic stress, which certainly has a role of its own in modern diseases, and importantly, the nutrition. And it has been shown that, for example, hunter-gatherers consume a diet which contains, on average, more protein and less carbohydrate than our modern Western diet. And especially carbohydrates seem to be important when it comes to cancer. That there exists a strong relation between carbohydrates and cancer has been known since the 1920s, when several physicians found that tumor cells would take up much more glucose and in turn secrete much more lactate than normal cells, even under normoxia. And normoxia means normal oxygen concentrations. This is therefore known as aerobic glycolysis or the Warburg effect, after Otto Warburg, who performed the most well-known experiments. Now, usually when there is normoxia, a cell will take up glucose and convert it through glycolysis to pyruvate, which will then be transported into the mitochondrium to yield citrate and fuel the TCA cycle and oxidative phosphorylation. And normoxia will therefore shut down lactate production. But in tumor cells, this mechanism no longer holds, and most of the pyruvate gets converted to lactate by this enzyme here and exported out of the cell together with protons. Now, the tumor cell has several advantages by having a high glycolytic rate. The first one is that it is independent of the actual oxygen supply in the microenvironment. And for example, aggressive tumors, they grow too fast for concurrent vascularization. So they will always have hypoxic areas in which they depend, or they have no other choice than to use glucose as a fuel. Also, a tumor cell no longer relies on intact mitochondria. And if they have dysfunctional mitochondria, that allows them to evade apoptotic signaling, because apoptotic signaling requires the presence of intact mitochondria. Now, a third advantage is that the lactate gets exported out of the cell together with protons, like I just said, and the protons will disintegrate the extracellular matrix and lead to apoptosis in normal stromal cells. And this allows tumor cells to more effectively proliferate and invade and metastasize. And the fourth important point is that glycolysis yields high amounts of phosphometabolites, intermediates, that can enter the pentose phosphate pathway to yield riboses for RNA and DNA synthesis and redox equivalents for fatty acid synthesis. And in fact, every fast proliferating cell, for example, activated T cells, they have a high glycolytic rate because of this, because they have to proliferate fast. Now here are some data by Eggert Holm and co-workers, which just show which quantify the Warburg effect. Basically, they have measured in 19 colon carcinoma patients during an operation the substrate uptake and production in tumors and in peripheral tissues. And what you can see here is that for free fatty acids and ketone bodies, there was basically no difference in uptake or production. But when you look at glucose and lactate, you see that tumor cells would take up about 30-fold more glucose than the peripheral tissue, and on the other hand, excrete about 20-fold more lactate. Now, it has long been a question what actually causes the Warburg effect, and Warburg effect is also so intertwined with all the other hallmarks of cancer that you can basically ask for the origin of cancer itself. And the picture that emerged during the last 50 years is that it is genetic mutations that give rise to hyperactivation of certain signaling pathways that will then lead to aerobic glycolysis, proliferation, etc. And especially these are caused by gain of function mutation of certain oncogenes and loss of function mutations of tumor suppressor genes. But during the last 10 years or so, a more complicated picture has emerged. And there are now many reasons to believe that these genetic mutations are not the primary cause of cancer, but they arise secondarily due to some other causes. And there seems to be a complex network of factors that can interact with each other and all give independently rise to tumorigenesis. For example, there are good reasons to believe that cancer can more be considered a disease of altered metabolism or a metabolic disease, like Tom Seyfried will tell you after me. In this case, defect or dysfunctional mitochondria are the primary cause and only secondarily lead to genetic mutations. They can also lead to chronic inflammation, and chronic inflammation by itself has been known for, I think, more than 100 years as a potent stimulator and driver of tumor growth. Similarly, epigenetic modifications, for example, silencing of certain tumor suppressor genes, can give rise to the malignant phenotype. Also, hypoxia by itself, and in aggressive tumors, we have hypoxic areas, will make sure that these tumor cells are dependent on glycolysis and they will upregulate their glycolytic rate. And a very important point is that hyperglycemia and hyperinsulinemia can both cause defect or dysfunctional mitochondria, and they can lead to chronic inflammation, which then stimulates all these processes of tumorigenesis. So it seems that genetic mutations are necessary, but not necessarily sufficient for cancer progression. And the microenvironment, for example, plays a very important role. And tumor promoters in the microenvironment include, for example, abnormal metabolism, an aberrant extracellular matrix, inflammation and fibrosis, the presence of growth factors like insulin and IGF-1, presence of certain hormones, mutagens, and reactive oxygen species. But the good news here is that many of these factors can be, in principle, countered by a Paleolithic diet. For example, a paleo diet is usually a diet lower in carbohydrates, and this by itself will lead to lower blood glucose levels. Now, blood glucose directly impacts insulin, and insulin indirectly impacts IGF-1, and both insulin and IGF-1, when they bind to their specific receptors, they will trigger a signaling cascade through PI3K, AKT, and mTOR, which leads to stimulation of proliferation and resistance to apoptotic signaling. Now imagine you have a cell which is on the border between going into apoptosis or becoming malignant. Then chronically high levels of insulin and IGF-1 in the microenvironment, like they occur in patients with metabolic syndrome, they might lead to favor the malignant transformation of the cell. Dietary restriction is also could be considered part of a Paleolithic diet, for example, in folks who do intermittent fasting regularly. And dietary restriction by itself will activate AMPK, which will then have a negative effect on the mTOR pathway. So let's go back one more time. I want to stress that this pathway is also frequently upregulated in many tumors, and this is part of the reason why tumor cells have such a high resistance to apoptosis and such a drive for proliferation. On the other hand, the hyperactivation of this pathway by genetic mutations renders tumor cells somewhat less flexible to the availability of nutrients and the metabolic pathways. They are more addicted to glycolysis. And therefore, the Warburg effect, or aerobic glycolysis, has been considered by some as cancer's Achilles heel and is now being explored for metabolic drugs, for example. And indeed, in vitro experiments have shown that cancer cells are very vulnerable when you deprive them of glucose. However, Jean just pointed out that in vivo, this effect might be questionable because by gluconeogenesis, you always make sure that your blood glucose level has a certain range or a certain physiological value. However, imagine a patient who has hyperglycemia, and what you see here is the oxygen gradient. So this is distance from the closest blood vessel, And you have here the oxygen gradient and the glucose gradient. It reaches fast into the tissue. Imagine the tumor, and here this area would be a hypoxic area. It's no longer reached by oxygen, but it's still reached by glucose. And it's well known that hypoxic areas are also the most chemo and radio-resistant ones. Now, if you're able to lower these patients' blood glucose level, then theoretically, at least, you could deprive those hypoxic cells from the blood glucose supply and eventually slow down the tumor proliferation and lead to necrosis or tumor cell death in this area. But even if this mechanism does not make a large difference in vivo, then we still know that there are some indirect effects of blood glucose. And we know this because, for example, starved animals, which have blood glucose levels on the lower physiological range, they survive longer and have slower tumor growth than their normal fat counterparts. We also know that, and Colin Champ had a nice poster yesterday about that, we know that hyperglycemia leads to elevation of insulin and free IGF-1. It leads to a state of inflammation or inflammatory signaling, and a competitive inhibition of ascorbic acid transport into immune cells, thereby blunting the immune response to cancer cells. Also, hyperglycemia, hyperinsulinemia, elevated free IGF-1 and obesity are all independent prognostic factors for cancer occurrence and poor survival in cancer patients. And it's important to stress that these are all hallmarks of the metabolic syndrome, and it shows that cancer patients have many things in common with those who have the metabolic syndrome. And this is because the tumor manipulates whole body metabolism. We know that in metabolic syndrome patients, the adipose tissue will excrete certain inflammatory cytokines. And in the cancer patient, these cytokines get excreted by the tumor tissue. Now, these cytokines will signal to the liver that there's some inflammation going on, and both liver and muscle will become more insulin resistant, even at a very early stage of tumor progression. Now, in this early stage already, the synthesis of glycogen in the liver and in muscle tissue is impaired. And muscles do either have a normal fatty acid oxidation rate or they have an upregulated fatty acid oxidation rate, in which case this will increase the rate of lipolysis from adipose tissue. Now, with more tumor growth as the disease progresses, we enter a late stage where the liver upregulates its gluconeogenetic rate, and the substrate for that comes either from more breakdown of fat from the adipose tissue, it comes from the lactate excreted by the tumor, and it comes from proteolysis, which occurs in muscle to yield alanine, the amino acid alanine. And this will be converted to glucose by the liver, further fueling the tumor. So this could be seen as a vicious cycle that develops. And the end result is that the cancer patient loses muscle mass and he loses adipose mass and turns into a cachectic patient. But this also implies that cancer patients need fat and not carbohydrates, because fat and ketone bodies, they will still provide a fuel for muscle cells, even in the cachectic state, while they can hardly be utilized as a fuel by cancer cells. It has been shown that if you increase the amount of fat in the patient's diet, this will lead to an attenuation of weight loss and fat-free mass loss, even if the patient's already cachectic. If you replace carbohydrates with fat, that will also lead to a lower glucose, insulin, and free IGF-1 level. And ketogenic diets may have additional special beneficial effects. For example, ketones have been shown to block glycolysis, they act neuroprotective, and they interfere with signaling pathways like the mTOR pathway. Some in vitro studies have been published that show direct anti-tumor effects of ketone bodies, like, for example, a study by Eugene Fine. And there's a larger amount of animal studies which show that animals on a ketogenic diet will survive longer, they have less tumor growth, they have less metastasis, they show a genetic normalization of the tumor tissue, and they have no loss of body substance, a normal weight gain if you use an unrestricted diet, and the animals are usually fit in contrast to their normal fat counterparts. Unfortunately, there's a lack of clinical studies. All we have are some positive case reports and preclinical studies. However, these studies give rise to hope that ketogenic diet is beneficial for the cancer patient. Positive effects that have been shown include weight stabilization or weight gain, an improved quality of life, diminished glucose uptake by tumors. Now, one study found that there were hints towards less proliferation of the tumor. A study done at our clinic by Schmidt et al. Showed improvements in many blood parameters, and you just heard the talk by Jean, who showed that ketosis correlated with stable disease or partial remission in his patients. So one last point that I want to stress is that when you think about doing a ketogenic diet for a cancer patient, it's important to not just focus on the macronutrients, but the quality of the nutrients. This can be learned from the Paleolithic approach, because in paleo diets, quality plays a larger role usually than just the quantity of macronutrients. For example, it's important that the omega-6 to omega-3 fatty acid ratio is not so high as it is in the standard diet, because more omega-3 will lead to more anti-inflammatory signaling, and omega-3 fatty acids have been shown to have some beneficial effects against the cancer cachexia. Usually a paleo diet contains no gluten and grain lectins. In this way you are able to avoid additional inflammation. You can ideally substitute with a lot of coconut oil, which has these medium-chain triglycerides that get readily converted to ketone bodies. And usually on the paleo diet, you don't have to worry about vitamins and secondary blood substances. And for example, from red wine, green tea, curcumin, broccoli, etc. And many of these substances have been shown, at least in vitro, to protect against reactive oxygen species and inflammation. And they are thought to have an epigenetic regulation of anti-tumor processes. So to summarize this talk, a paleo diet, and unfortunately I wasn't able to go into other aspects of a paleo lifestyle, but they have similar mechanisms like exercise and vitamin D. So they can contribute to cancer prevention through reduced inflammation, normalization of blood glucose, insulin, and IGF-1 levels. They positively affect body composition, and they have an epigenetic suppression of tumor-promoting factors. And the same principles might improve the prognosis of cancer patients, whereby ketogenic diets could be especially effective. Thank you.
One question.
Hi. I have a girlfriend, she's 35, and she's looks very healthy. She's a dance instructor and stuff, and she's been a vegetarian her whole life, and she's just, been diagnosed with brain cancer. Yeah. So I'm just wondering if it's possible for her to do a ketogenic diet if she's vegetarian, like just do all fats. Is that still something that can help her?
I think so. I would try it. So, I mean, you can derive most of your calories from fat. You should derive most of your calories from fat. And this does not necessarily have to be animal fat. Coconut fat and palm fat are very effective for stimulating ketosis.
Mm-hmm. Okay.
So,
and because right now she's following like a alkalizing diet.
mm-hmm.
Is it just adding a lot more fat to that her vegetarian diet with that? I don't know if grains and stuff are part of that for her, but is that could help maybe?
I would make so I would make sure that she is in a ketotic state. I think that's important.
Okay. Okay.
So it depends. Some people can eat as much as 60 gram carbs per day and be in a ketotic state. For some people, it's only 20 grams. That's individually different, but
Okay.
yeah.
Thank you. Sorry.
And I think the next talk will address exactly that.
Okay. Thanks.
Yeah.
Thank you, Dr. Clement. We'll